So here would be an embryo with these inner cell mass, and then can you take out a stem cell and grow it? And this was something that scientists tried for a really long time to do, but every time they got these cells out, they would die because they had not figured out the culture conditions that would work. So back in 1981, two groups, so Marty Evans and Gail Martin, both in the same year published that they, for the first time, were able to establish mouse embryonic stem cell lines. Now, these were stem cells that they'd taken out of a blastocyst, and they found the exact culture conditions that would allow them to maintain these indefinitely. So now you have stem cells that are maintained as stem cells growing in culture in the lab, and you can start to study what is it about them that gives them this amazing pluripotency. The next development in terms of embryonic stem cell lines took 17 years to come about, and that was work done by Jim Thompson's lab, and his was the first lab to describe human embryonic stem cells. Now this is taking cells out of a blastocyst from a fertilized human egg and growing them in culture. Now once he did this, then there was a huge controversy because this was the destruction of a human fertilized egg. These were eggs that would have been discarded anyway from fertility clinics. But this breakthrough led to George Bush banning embryonic stem cell work in the US, at least any research funded by federal money, which is most of it. President Obama, 8 years later, did reverse that ruling, and the stem cell community has started to recover from that. But this is a controversial field because you're dealing with the very early beginnings of an organism